All right, so this video is for section 7 of chapter 8 in biology. So we're going to talk about different types of mutations. So your objectives, we got to distinguish between those different types. We got to explain why mutations may or may not affect a phenotype, and we got to list some factors that can cause mutations. So first off, obviously a mutation is a change in an organism's DNA. A point mutation is when one nucleotide is substituted for another. And there's actually two different types of point mutations, and it's only easy to understand this if you can actually see an example in action. I'm going to give you these examples in here. You don't have to try and draw these in your notes because it would be difficult to do so, but just try and follow along because each one's a little bit different than the next. So all these pink pieces of mRNA are different from each other in one small little way, but those little changes can make big differences in the long run. So you just need to follow along so that you can understand how this works. The first one we do is just normal, right? I'm assuming there is no mutation. So I'm going to start at a start codon, and I'm going to start reading from there, and I'm just going to get what I get, right? So AUG is a methionine, GAU, got to look it up real quick. That's ASP. AAC is an ASN. UAC is a tyrosine, and then CCG is a proline. UAA is a stop, so I'm done with this protein, right? These amino acids will be linked together by that ribosome. Again, this is probably technically a polypeptide, but those words are pretty much interchangeable for you guys. But this is what the protein is supposed to look like, right? So let's see what happens with some mutations. Again, the first type is a missense. I'm supposed to only change one amino acid. And if you look at this line versus this line, the one difference is that I put a A here instead of a U, right? Which if that's going to happen, it could be a mistake in making the RNA. But if it's going to be a mutation, it has to be something that lasts, right? So that means that I couldn't have had an A here in my DNA. It must have been a T. So some damage happened and that base was replaced with a different base. That A was replaced with a T. And then that's permanent. That's in that DNA forever, and it's going to be passed on to future generations. So if that A got swapped out for a T somehow, I wouldn't get a U here. I'd get an A instead in my mRNA. And understand that most of this remains the same. I'm still going to get a methionine. I'm still going to get an ASN here. I'm still going to get a tyrosine. I'm still going to get a proline here. But this base change, that U to an A, is going to make a difference. GAA is actually going to bring in a GLU instead of an ASP. So I have a slightly different protein. Now understand, this may make a big difference or it might just be a small difference. In this polypeptide that we made that only has 1, 2, 3, 4, 5 amino acids in it anyway, that's actually a huge difference. It's going to matter that that thing got switched out. But if you start thinking about proteins in your body that could be like 4,000 amino acids long, one little change might not actually make all that big of a difference. It depends on where it happened and what you changed for what. But this kind of mutation doesn't necessarily have to be debilitating. It doesn't have to be a terrible thing that happens. It could be just a small little change that all of a sudden you have red hair instead of brown hair, right? So who knows what the effect is after the fact, but this is what a missense mutation looks like. So if I change one nucleotide, right? Remember I changed that A for a T in the DNA, right? Or vice versa, whichever one it was. Yeah, it was an A for a T. And it changed the resulting RNA and changed one amino acid in my resulting protein. That's a missense mutation. It's a type of point mutation, but it's missense because it only changed one amino acid. So aside from missense, there's also nonsense mutations, and those are a little bit different, right? My third one here is supposed to be a nonsense mutation. So again, I got codons here that may or may not be the same that they were before. Now the one little difference this time is actually over here. So I'm gonna highlight that real quick. Let's just use orange or something. So instead of having a C here, I had a G. Now remember, I changed this one before. That's actually the same now. It's a U just like it was before. So we're assuming that T isn't a problem anymore. But in order to get a G here instead of a C, that means this time the mutation would have had to have been a C instead of a G in my DNA up here. Again, it has to be in the DNA, not in the RNA, the mutation, or else it wouldn't be permanent, and it wouldn't really be a problem. But if this was a C up here in my DNA, then I wouldn't get a C here. I'd get a G instead, like I did down below. But what that does is now I have methionine, I still have ASP, I still have ASN, but UAG is actually a stop codon. Instead of bringing in this tyrosine and then following it with a proline and then stopping, I stopped early. So now I just have this 3 amino acid chain that's going to be made. And again, probably not the best thing for this small polypeptide to lose 2 amino acids. That actually does make a pretty big difference. But imagine if this protein was supposed to be 4,000 amino acids long. There's a really big difference between 3 amino acids and 4,000 amino acids. So nonsense mutations are typically really, really bad, right? If you bring in an early stop, and again, it happened because of one nucleotide change. It's still a point mutation. All I had to do is change one nucleotide. I changed a G into a C, and all of a sudden I have this early stop. And if this was supposed to go on for a long time and make this big old protein that's really important for me to be alive, I might not be alive anymore, right? So early stops are typically really, really bad. But that missense mutation that we had before, where only one amino acid changed, those may or may not matter, right? They can actually be a big deal, but more often than not, it's a small change that doesn't really affect the protein very much, especially if it's a big protein. If it's small polypeptide like this, it's still a problem, but you can understand what I mean. So then finally, aside from point mutations, you can also have frame shift mutations. These ones are a little bit harder to visualize, but this is involving the insertion or deletion of a nucleotide. These types of mutations are really bad, generally speaking, because almost all the codons are going to be affected, or at least every codon following said mutation is going to be affected. So again, if I go back to, remember, I'm not supposed to have these changes permanently in my DNA. This last strand is different a little bit than the first one, but again, this one's bad for a reason, right? So if I try and figure out what my little difference is here, right, it looks like, compared to the first one, that I'm missing something here. Right here, I was supposed to have an A, right? And what that means is that, let's say my DNA, my original DNA, had a T in it, but what if there was damage here and all of a sudden A is connected to G and it just skips right over T? What if I lost that base? So that could happen, but what it does is it causes a frame shift, and that's going to cause serious problems down the line, because I'm still going to read an AUG, and I'm still going to read a GAU, and then I'm going to have an AAC, but then the next one is going to be a UCC instead of a UAC, and then my next one is going to be a GCU instead of a CCG, and then my next one is going to be a AAA, and then my next one is going to be whatever it happens to be. This could be a much longer piece of mRNA, right? So what you're supposed to be able to see here is that I actually can see these codons, this AAA, I can see that up here. It's just not what I would have read originally, right? This G or CGU, that's up here, GC or CGU, and then the other one would be it's a UCC because that A is missing, it disappeared, right? So when I say frame shift, what I mean is instead of reading this black frame here where it was CCG, I'm reading this red frame here where it's CGU. And what happened is I skipped over my stop, and who knows how long I'm going to go on this one before I find a stop codon. So again, it's not just going to change one amino acid. It's going to change an initial codon, obviously. Again, my UAC got turned into a UCC. But then every single codon after the fact is also affected. So the big problem here is every codon after that insertion and deletion is going to be affected. And that means that basically all of your protein is going to change, and it's going to go on forever because it doesn't stop when it's supposed to stop. So you make this really big protein, because before I was only supposed to have 5 amino acids, and now I might have 16 before I hit a stop. I might have hundreds before I hit a stop, right? So the protein is going to change drastically, and this is almost never good, right? Not likely to be a good thing at all. So there's a difference between missense, where I just change one amino acid for another, nonsense, where it stops early, and any kind of frame shift mutation, right? Missense and nonsense are typically the worst types of mutations. Missense ones maybe don't make much of a difference. So again, might have said that wrong, I'm trying to think. Missense, not usually a huge problem, but nonsense and frame shift mutations are usually pretty bad, right? If you stop too early and it's supposed to be a much longer protein, that's a problem. Frame shift, on the other hand, it's very unlikely that they would stop on time, right? They'd be way too long, right? Whereas missense, you only change one amino acid. Changing 1 in 100 might not make a big difference. It can make a big difference, but it's not as likely to. Right, so those are the different kinds of mutations.